In this part of our video seminar series, I'll take something of a detour from the details of our mitochondrial temperature measurements. Instead, I'll review some of our earlier work on Drosophila, which suggests the importance of mitochondrial heat production in the biology of the fruit fly. Notably, Drosophila is a poikilotherm, an organism whose body temperature has hitherto been presumed to be largely dependent on that of its external environment. To kick off, I'll just recap the main points from part 3 of our series, where we considered evidence that mitochondrial temperature responds homeostatically to a wide variety of stresses, including substrate availability, ATP demand, metabolic insults, and thermal shock. In this segment of the series, we'll focus on the fruit fly Drosophila, and our past work indicating that mitochondrial heat production may be an important factor in a number of processes, notably spermatogenesis, cell migration, proteotoxicity, cold resistance, and possibly also in development. The biological consequences of elevated mitochondrial temperature are extremely diverse and impactful, and this will be the topic of this and indeed all of the remaining parts of the series. They include impacts on mitochondrial dynamics and homeostasis, apoptosis, immunity, and heat delivery both within and beyond cells, as well as aspects of disease and evolution. I will address these issues in parts 5 through 9 of the series. For now, since this presentation is on Drosophila, I think it's probably helpful to say just a few words about the fly's life cycle. Although a complete description of Drosophila biology and of the vast array of molecular tools now available to study and modify its genome with predictable effects on development, metabolism, and physiology is obviously beyond the scope of this series. The complete life cycle from egg to adult takes about 10 days in flies that are cultured at 25 degrees Celsius. The adult flies living typically between 40 and 90 days, depending on strain. After the 3 larval instars, the major feeding and growth phase, the flies undergo a complete metamorphosis as pupae, wherein cell proliferation and differentiation of rudiments set aside earlier in development take place. And the organs and appendages of the adult fly are formed, which are essentially the same as those of mammals, except that the skeleton is external, not internal. We also need briefly to revisit the alternative oxidase, the enzyme found in most organisms, but not in vertebrates nor in insects, that can substitute for complexes 3 and 4 of the mitochondrial respiratory chain if either of them is inhibited or overloaded, but note that it does so in a way that does not contribute to mitochondrial membrane potential and is therefore purely thermogenic, at least in terms of energetics. So, wherever it's active, AOX changes the energetic terms of trade of mitochondrial metabolism, releasing more heat and making less ATP than the full standard OXPHOS system would without its intervention. We transferred a cDNA encoding AOX from the tunicate Siona into the genome of Drosophila, creating 2 versions, one subject to regulation using the binary GAL4 UAS system, the other constitutively expressed under the alpha-tubulin promoter. AOX flies were healthy and essentially indistinguishable from wild type, even when the transgene was expressed at a high level. However, when we looked more closely, we identified a few phenotypic deviations that seemed interesting. Ubiquitously expressing AOX males were fertile and able to mate, producing normal numbers of offspring. However, their sperm was routinely outcompeted by sperm from wild-type males in the classic sperm competition assay. The total amount of sperm produced by AOX males was much less than from wild-type males, and the orderly process of spermatocyte differentiation was spatially disrupted inside the testis. We traced the site of highest AOX expression to the pigment cells of the testis sheath, with almost no expression in the spermatocytes themselves. Our working hypothesis is that in the wild-type fly, electron flow through complexes 3 and 4 in these cells is physiologically blocked, thus decreasing greatly the overall production of heat in the testis sheath and ensuring a cool environment for sperm maturation, which seems to be essential in many animals, including mammals, although the biological reasons for this are not well understood. In AOX males, however, the respiratory bypass restores electron flow and heat production even when complexes 3 and or 4 are physiologically inhibited. That heat production is even greater than it would be in the wild type were complex 3 not unavailable, due to the thermogenic nature of AOX-mediated respiration, thus warming up the local environment and disturbing sperm production. This concept is supported by the recent work of others, showing that continuously culturing male flies, wild-type male flies, at elevated temperatures, such as 29 degrees, essentially produces the same effect as AOX on the reproductive system. Switching to the topic of cell migration, we and many others have observed a classic cell migration phenotype in flies, cleft thorax, due to the failure of midline closure during pupal development and resulting from disturbed JNK or steroid signaling. However, we found that AOX expression was able to alleviate this phenotype and also facilitated cell migration in an analogous mammalian model, the classic scratch assay, which was further enhanced by antimycin, an inhibitor of complex 3, but not other drugs, consistent with the effect being thermogenic due to AOX. Cell migration has elsewhere been shown to be stimulated by heat. A further phenotypic impact of AOX expression is to mitigate the pathological effects of the expression in flies, notably in neurons, of proteins that form pathological aggregates, at least in humans. One example is the Alzheimer's disease-associated amyloid beta peptide A-beta, which, in its most severely pathological form, is also highly toxic in flies. AOX doesn't provide a complete cure, but does double the extremely curtailed lifespan of A-beta expressing flies. In mammalian cells, AOX also repressed the formation of the A-beta peptide. Furthermore, AOX also blocked the pathological effects of a different aggregating protein in flies, although this has not yet been published. From cell models, it is already established that heat and heat shock proteins can help resolve such aggregates, although the relationship is not straightforward. This all leads to the challenging hypothesis that proteotoxic disorders in humans may actually be caused by insufficient mitochondrial heat production, though obviously that's only a hypothesis at this point. AOX expression also facilitated the recovery of flies from cold-induced coma. AOX flies almost all recovered from a night in the cold room after 45 minutes at room temperature, when hardly any wild-type flies had become active over that time. Although we don't know for sure that this was a thermogenic effect, it's obviously one plausible, at least partial explanation, although at this point it's only a hypothesis. AOX conversely had a negative effect on the completion of development of flies reared on low nutrient medium, and the effect was temperature sensitive, although the metabolic effects are complex, and heat production at the expense of ATP may or may not be in play. On the other hand, AOX expression accelerated the development of flies reared on standard medium in the cold. Although recent calorimetric, enzymatic and metabolomic studies from the lab of our collaborator Marcos Oliveira in Brazil showed that AOX expression diverts metabolism into a more proliferative or biosynthetic program, rather than acting thermogenically. A note of caution, therefore, in interpreting all experiments involving AOX. To investigate the effects of AOX and other manipulations and stresses on heat production and heat transfer in the whole organism, we need the approach outlined in part 2 of this series, in which the ratiometric fluorescence of 2 fluorescent reporters is used as a temperature readout in different organs, as well as in different developmental stages, cell compartments, and so on. Creating such tools is a work in progress of the Tampere team. To preview the material that we'll cover in the next edition of this video series, we'll be taking a deep dive into the implications of elevated mitochondrial temperature and heat production for cellular processes, such as mitochondrial fragmentation, fusion and motility, apoptosis, or programmed cell death more generally, and immunity, as well as considering briefly some ideas regarding the identity of possible heat sensors in cells and in their mitochondria. It's said that programmed cell death is one of the few cellular processes visible from outer space, as the deciduous trees of the world's great forests shed their foliage in autumn. Finally, to close this segment, I'll acknowledge the support of my funders and colleagues who did most of the work described, notably Danielle Fernandez Ayala and Kia Kemppainen, who created and studied the AOX flies, as well as Anna Angelkovic, Sina Sari, Marcos Oliveira and Eric Dufour, who all contributed in different ways to the work I've surveyed here, as well as Riyad El Khouri in Paris in Pierre Rustin's lab, plus the sources for various photos and illustrations that I've used.